try again. Welcome to the not online session of molecular biology. Crazy weather we've been having here. That does remind me, however, if there's anyone who has trouble getting access somehow to YouTube, seems hard in this day and age, but let me know. So if there are some internet issues and so on and so forth, I know that not everyone has access to internet, and I think that's what happened last night when I tried to upload the lecture, because I could have sworn I uploaded it, and it had started uploading, but evidently it didn't completely go through. So that's that. I do have all of your Scantrons here. I'll try and finish a couple of minutes early so we can give you a chance to take a look at those. Hopefully everyone's compared their exam to what they thought they had on the Scantron. If there's any disconnect as far as that's concerned, please let me know. I've got scanned versions, like optical scanned versions now, of all of those Scantrons. So if there's something that just got misgraded somehow, please let me know and I will correct that. Unless it's in your favor, then don't tell me. Or, well, I guess it's up to you if that's the case. So that being said, you may have noticed that I didn't put in clicker questions on the lecture that I had last time, so I could do some now. But if people have opinions about that, I could have done that. I was sort of half, should I have clicker questions in here or not? A lot of it's so that I can get that feedback rather than anything else. So that being said, what are we going to do? Clickers. Yay. You guys are so well trained. This is great. So you've discovered a new and rare DNA damage that covalently adds a benzene ring to adenine residues in double-stranded DNA. What repair mechanism is it most likely to be used to repair it? Mismatch repair, base excision repair, nucleotide excision repair, non-homologous end joining, or homologous recombination? And again, feel free to talk about this. Just the idea. Four. So let's see what we think. We think bit of a difference of opinion here. We seem to have basically three options. Nobody liked non-homologous end joining or homologous recombination, which is good because it has nothing to do with double-stranded breaks, which is what we'll be talking about for most of the rest of today. Then we've got mismatch repair, base excision repair, nucleotide excision repair. So three different mechanisms for dealing with things. If you've added a benzene ring to an adenine, does it look like an adenine? Okay, what does base excision repair do? It cuts out, mismatch repair. Mismatch repair is, what do we see with mismatch repair? Mismatch repair is if it's the wrong one of the normal nucleotides. One of those gets taken out. So it's got a benzene ring on it. It's a normal nucleotide? Definitely not. So what about base excision repair? Cuts out a single base. What's really very important about base excision repair? How does that base get cut out? Gets cut out by a specific kind of enzyme, which is a glycosylase. So enzymes usually are extremely specific in terms of what they deal with. So we talked about, well, I talked about anyway, uracil DNA glycosylase. Takes uracil when it's in DNA and chops it out. So if it's a specific kind of mutation that is common, then you're likely to have base excision repair because you'll have a specific enzyme that takes it out. However, if it's something which is rare, what kind of mechanism are you likely to use? Nucleotide excision repair. Why nucleotide excision repair? Because it's looking at the overall structure of the DNA. What would happen if you put a benzene ring in here? It's going to really screw up the structure because of all the base stacking interactions that you have in there. So it's really going to mess with the structure of your DNA, and it's rare. So what do we think? C. Yes, and I would agree with you that C is the correct answer for this. And just because it's so much fun, let's do another one. Seek with multiple choice. Go to our next one. What's the template used by the telomerase enzyme TERT to make telomeric DNA? The leading strand DNA, the complement of the leading strand DNA, the lagging strand DNA, the complement of the lagging strand DNA, or the telomerase RNA? Ten, 5, so I guess a lot of you didn't make it to the biology seminar last Thursday? No? Yes? Anybody actually there? Yes? It was really cool. So this is a potential job candidate in the biology department, and he works on telomeres and telomerase, and particularly what aspect of it? The telomerase RNA. So I guess I did not do a very good job of talking about this last time, or you had trouble again finding the lecture. So the telomerase enzyme, and to some extent, if you know what the TERT stands for, it's telomerase enzyme reverse transcriptase. So what do reverse transcriptases do? They take RNA and make DNA copies from it. So the template that the telomerase uses is an RNA that it brings with it, which is really unique in terms of enzymes, certainly in terms of polymerase enzymes. I don't know of any other enzymes that bring their own template with them. And it's a relatively short piece, and that's what continues to get copied. And so, a couple of important things here is A, how does the telomerase work? The second one is really about the template. And so template is also kind of review as far as replication is concerned. So it's the nucleotide sequence that's getting copied on the opposite strand. Now, one of the things that you can think about, and maybe it'll show up on an exam, who knows, I haven't written it yet, is what the primer would be for the telomerase enzyme. Yeah, I had a question. Sorry. Question?
It's okay, I got an answer.
Okay. So I sit next to smart people. Good idea. So yes, the answer here is E. One of the things that I also wanted to talk about here briefly was a little bit about the telomeres and the telomerases, having to do with this kind of structure. Hopefully, it's clear why you need a telomere. Why do you need a telomere? So that when you have replication, it doesn't get shorter and shorter and shorter. Because for each of these lagging strand pieces, the semi-discontinuous replication, you have an RNA primer. That RNA primer gets removed. Normally, it would be copied due to the DNA polymerase that would sit down here and extend it. Of course, there's no 3' OH over here. Nowhere for it to be extended from. So how do you deal with that? The way that you deal with that is you actually extend this strand. You extend the template strand. And that's what the reverse transcriptase part of the telomerase enzyme makes. It makes a template strand so that the normal DNA polymerase machinery, together with primates and everything else, can copy in that opposite strand. You're always going to end up with this little 3' end overlap here. Always is going to be a single-stranded piece because in any case, you're always going to have your primer, RNA primer, that gets removed. So that little end piece has to be protected. How is it protected? Anybody? T-loop. It loops around. It loops around in base pairs back to some of the repeated sequences that happened before that. And there are lots of other proteins that are involved with this telomeric structure. Shelterins, etc. I don't expect you to remember all the names of them. People seem to have different names for them in different organisms, and that was also part of the talk that we heard on Thursday, that different organisms have different sequences in their telomeres and different proteins that are associated with them. So protection, just from when you replicate, you have a couple extra copies at the end. The other thing is that these ends, and particularly 3' extensions, are actually incredibly recombinogenic. And we're going to talk about homologous recombination here. It's all about extending 3' ends. And so one of the other things that's really important about the proteins that are associated with these ends is so that the telomere doesn't go and hook itself up to another chromosome and end up fusing chromosomes. You remember those crazy cancer chromosomes that we saw before? That's actually one of the things that happens, is you have the telomeres fuse and all kinds of craziness happens with your karyotypes. One of the things I'll do after lecture, I'll link a really nice talk by Liz Blackburn, who got the Nobel Prize in, I think it was 2009, for the discovery of the telomerase enzyme and RNA, who was given a talk on eye biology. Anybody know eye biology? Yeah, so there's some really nice seminars there. It gets into more detail than we cover in this class. But usually these are the people who've done a lot of these experiments who are involved there. So I'll add that reference into the discussion notes as well. Again, last time, sitting in front of my computer, having my daughters go, what the heck's he talking to? About how DNA is actually a really crummy molecule, but really critical molecule in terms of being pretty unstable, in terms of living in an aqueous environment at relatively elevated temperatures, and us liking oxygen. All kinds of things can happen to your DNA molecule in that process. All of the genetic information is chemical information. Any kind of chemical changes are going to be those which could actually change the genetic information, which is usually a bad thing, but if you didn't have any changes, we'd probably still be slime. So you have to have some changes and some evolution actually taking place. So it seems to be a balance, and probably why we've evolved to use DNA as a genetic material. Stable enough, lots of repair mechanisms, but also having some changes. So we talked a little bit about this already, just wanted to review it briefly, the mechanisms of the base excision repair and nucleotide excision repair, which are really the sort of the main mechanisms that are used most commonly, and the main mechanisms as far as we'll talk about in this class. There are a few other mechanisms too, but we won't talk about those in detail. So base excision repair for very specific changes and very common changes that happen to your DNA, because you need a specific glycosylase. So here's that specific glycosylase. It binds to and cuts out uracil in the DNA. Uracil's in the DNA because your cytosine has been deaminated, which happens again spontaneously quite frequently. Once you've removed that through the glycosylase, now you have your DNA that's missing this one base right here. That now is an AP site. Apurinic, apuridymic. Why did they just call it an abasic site? I don't know, but it's known as an AP site. AP sites also happen spontaneously, and this seems to be the most frequent kind of DNA damage, just by depurination, which happens. The purines fall off, so adenines and guanines just fall off of the sequence. So once you have one of these AP sites, Then there's an endonuclease and a phosphodiesterase, which you could also call an exonuclease. So endo cuts in your DNA, exo chews up, in this case, just one nucleotide. Now you have a gap, a 3' OH, a template. DNA polymerase can do its job and fill in this particular single nucleotide that was taken out. So that's you have specific changes. Again, it's really specific to that glycosylase if you have a modified base, and also just depurination, that matter depyrimidination, much rarer but also happens, will give you those AP sites. Questions about base excision repair? Okay, so that's, again, very specific kinds of changes. More general kinds of changes, and often really big kinds of changes. So pyrimidine dimers, what happens here is you have two pyrimidines, usually thymidines, but not always, that are right next to each other in your DNA sequence. Presence of UV irradiation, they rearrange their benzene-like rings, conjugated ring structures, and stack these two bases directly on top of each other. And to do that, you have to twist the DNA. And I'm going to come up, take a look at the structure, because that cyclobutane ring is a planar structure. And the only way to get a planar structure at the bases that are offset relative to each other is to line those bases right on top of each other. As soon as you do that, you've messed with the structure of the DNA, the double-stranded structure of the DNA. And this is true for all kinds of big changes that happen to the DNA. So since a big change is happening in the DNA, what happens is you chop out that big change. But instead of chopping out just the little piece, you chop out a much bigger piece. And so strange things happening with the structure of our DNA. We make some cuts on either side of that. These are now endonucleases, because again, they're cutting inside the DNA. DNA helicase is required to get rid of this piece, unwinding that particular piece of DNA. Now you have a 3' OH, a nice template, DNA polymerase, DNA ligase come in and repair this whole piece of DNA. So, two major flavors of repair. Turns out that this nucleotide excision repair should seem really familiar. Why? It's actually really similar to base excision repair. You make two cuts. Sorry, not base excision repair, mismatch repair. Eventually I get that right. Mismatch repair. Mismatch repair, because you've got the wrong base here. It's the wrong base pair, but there's not a specific glycosylase. If you had a specific glycosylase that's cutting out all your thymidines, you have a real problem. So again, it's the structure which is being recognized as being wrong because you're trying to make these base pairs that don't fit properly. And then that can get cut out by a very similar mechanisms. And cuts on either side of the mismatch, helicase it gets rid of it, resynthesized. So extremely similar process. Yeah?
Does it require the mix in the same way that the mismatch does? And if so, does it, for the bacteria, does it create the mix?
Okay.
Does it recognize the section that it's going to deteriorate?
Yeah, so the question here, and sorry, I'll paraphrase it, has to do with the mechanism, exactly what's going on here. And so this, and you asked about the NICs, that you can also consider as being these exonucleases. So they're coming in, they're cutting the DNA backbone. So it's extremely similar. In the case of mismatch repair, particularly in eukaryotes, those NICs are already present on that strand. Because it's often the lagging strand and discontinuous. You're having the pieces that haven't been quite filled together yet. So you don't necessarily always have to make two NICs. In the case of nucleotide excision repair, you're always making two NICs. But also for mismatch repair, say in E. coli, where you've just got a methylated strand and a non-methylated strand, you're going to be making NICs, or these endonucleolytic cuts, two of them on either side. Yeah?
Can we say that nucleotide excision repair is for things that affect more than one base, while base excision repair affects just one, or is that too much of a generalization?
Okay, so the question is, and it's a great question, is nucleotide excision repair always used for changes to more than one nucleotide? And the answer is no, because anytime you've done a particular change that changes the structure, and in the case of mismatch repair, it's just a single base, which is that change. It's just that it's looking at the structure in general of the DNA. And that could be just like the clicker question that I had, adding a benzene group to one particular base which is there. The base excision repair, which again, you'd think, okay, single base change, you use base excision repair, but that only works if you have a specific glycosylase. And evolutionarily, you're not going to have a specific glycosylase for every single one of the possible changes that you could have to a nucleotide. Yeah?
The excision nuclease, does it have a defined pattern that it has to know to find in the DNA to know where to put those nicks?
Okay, so the question is there a specific sequence involved? And this is paraphrasing your question. For this excision nuclease, since it's just looking at structure, no, it's not. But one of the things that if you count the number of bases around here, you'll notice it's almost exactly 10 nucleotides. What does that mean? It's going to be on the same side of DNA when it actually makes those cuts. So what first has to happen is you need to have a protein which recognizes there's a problem with the structure, and then that brings in the nuclease, which is going to be on either side of that protein that notices that there's a problem. And it actually gets back to, some of you may have noticed right at the beginning of lecture, I had a sort of 3D looking image up there. I'm actually heading over to the engineering department to get those 3D printed, and that particularly was a model of the mut-S protein interacting with the DNA. So exactly that recognition protein in terms of interaction with the DNA and that structure. Okay, other questions about this? Because I wanted to spend more time on this again since it was a remote lecture, as far as that's concerned. Okay, so the other thing that I talked about on Friday, was it? I guess it was Friday, was double-stranded break repair. Double-stranded break repair is a real problem as far as DNA is concerned because you don't have your backup or redundant copy. You don't have the other strand of your DNA. So double-stranded breaks, you have to figure out a different way of dealing with those. And it turns out, two major different ways that this happens. There's the quick and dirty mechanism, which is non-homologous end joining, and the nice, precise mechanism that only works if you have a homologous copy, which is usually very, very similar in sequence, which is there. Non-homologous end joining is pretty straightforward. Again, you have this cut which has happened somehow, ionizing radiation, and we'll take a quick look at some of the other ways you can get double-stranded breaks. It's actually happened quite frequently in DNA. Non-homologous end joining, you have proteins that bind to the end of this, exonucleases that chop in at the ends. Then you end up with blunt ends. They get put back together. Now, what happens? Any kind of exonuclease here is going to get rid of those bases. So if there was any important genetic information there, it's now gone. So quick, yes. Dirty and potentially problematic if that happens to be an important sequence right in there. Yes, in the back.
Does the relative number of protein coding genes space out so that if this does happen, it reduces the likelihood of that it's going to affect a function, a necessary function?
So the question again, just to repeat it so people have it here, is are protein coding genes, or you could also say the important sequences, spread out stochastically such that on average, you're going to have fewer and fewer of these? The answer is actually no. It seems that genes have been kind of thrown together willy-nilly. So the fact that there's only 1.5% of coding is a potential good thing as far as this is concerned because you're not as likely to hit them. But it does turn out there are regions of the genome which are pretty closely packed with genes, and that's where we actually see a lot of the genetic problems, the genetic defects that happen because of these kinds of repair processes. So it turns out that some genes are more susceptible to mutation than others. These are also what are called some fragile sites. We end up with many more mutations than you do in the rest of the genome. Yeah?
So just like the interaction of like transcription and stuff, like does that make it sort of more likely that double-stranded break is going to happen or something?
Okay, so the question, I guess, sorry to paraphrase your question here a little bit, has to do with, maybe has more to do with repair, if I can turn around your question a little bit. Are regions of the genome that are being actively being transcribed or replicated going to be repaired quicker or not, as the case may be, or are they going to use homologous recombination versus non-homologous end joining? Is that more or less your question?
I guess that was just more like just the activity that's going on because it's a gene coding site or a protein coding site. Does that make it more possible or likely that something is going to happen?
Okay, so basically the question is, if it's an actively transcribed or replicated gene, is it more or less likely to be damaged in this kind of way? The answer is apparently not so much for double-stranded breaks, but definitely for some of the base excision repair and nucleotide excision repair, because some of that machinery actually comes along with the RNA polymerase. So the RNA polymerase itself also is associated with, and we talked a little bit about transcription coupled repair. I think I did on Friday. I hope I did. Go back and check my notes. But yeah, I'm pretty sure I talked about it. So you do have, and it's specifically those nucleotide excision repair proteins that are together with the RNA polymerase. So if it runs into something which it can't get through for some reason, that'll get repaired faster than some change somewhere else in the genome that's not actually being actively transcribed and then made into protein. But for double-stranded breaks, that seems to be less common. However, on the other hand, as far as replication, it turns out that replication is often causing double-stranded breaks. And so that's a great lead-in to this slide, which I talked about really briefly, but wanted to get back and talk a little bit more about. DNA replication, if you have a nick or a cut on one of the strands, if you replicate past that, then you've generated a double-stranded break. And so that's just shown here. Here's a cut in one of the strands of DNA. Your replication fork comes through here, and you end up with a double-stranded break. This piece is not connected here anymore. In fact, I wondered, how are you having all these nicks happening? If you think about topoisomerases, particularly topoisomerase 1, what does it do? It nicks the DNA and allows it to spin around and then puts it back together. Well, if it doesn't put it back together, you have a nick there. And if the polymerase is coming along and gets to where that topoisomerase was before it's had a chance to religate, that's going to generate a double-stranded break. And it turns out that there are a lot of really good chemotherapy drugs that block topoisomerase 1 for exactly this reason. So that whenever you're replicating through here, and cancer cells are replicating really rapidly, they'll end up generating a whole bunch of double-stranded breaks. So topoisomerase inhibitory drugs are actually really good at making double-stranded breaks, which I wouldn't have thought of till I actually watched one of these iBiology lectures yesterday. And they mentioned that this is actually probably one of the more common reasons that you have this. Yeah?
Does that target the cancer cell replication specifically, or is it just all replication and they just try to localize it?
Yeah, so the question is, does that target cancer cells specifically, or does it target all cells? And the answer is, if it's a topoisomerase 1 inhibitor, topoisomerase 1 is the same in cancer cells and all the rest of the cells. It's one of the reasons that chemotherapy makes you sick. So it actually affects all of these. However, it's those cancer cells that are proliferating really rapidly. So they're undergoing a lot more DNA replication than other cells. So it's definitely possible that, again, particularly if you're giving systemic chemotherapy drugs, which are topoisomerase inhibitors, that can be a big problem. Yeah?
So after you get the double-stranded DNA, the break, what happens then? Like you get the
Perfect lead-in to the rest of the slide. Thank you. So yeah, what happens next? So what happens next? What happens next is you have a exonuclease that is a 5' to 3' exonuclease that chews up one of these strands, leaves you with a 3' overhang that should sound really familiar because we just talked about it from the telomeres. That 3' overhang then undergoes a process called strand invasion that we'll look at in a lot more detail in the next couple of slides. Strand invasion now allows this single strand to base pair over here with this strand, which now gives it a primer and a template so it can continue and replicate. This strand invasion generates what's called a D-loop or a displacement loop, and that's because one strand's been pulled off of the other strand and is now replicating over here. This now will extend. You end up you've got another nice little lagging strand over here. You do have to deal with this, and we'll talk about this crossing over piece in just a second. It's called a Holliday junction. So it's that double strand becomes single strand, strand invasion, extension, Holliday junction, and you end up with a normal replication fork again. This is probably how most double-stranded breaks happen in cells, is that you have this nick in one strand, template, that generates your double-stranded break, and then you restart from there. The same process can be used for repairing regular double-stranded breaks, which are due to some kind of ionizing radiation, some other kind of double-stranded break that happens. And it turns out in meiosis, it's actually a specific double-stranded break which is made by the cell. It makes a double-stranded break in order to get this homologous recombination to take place. So here we have our double-stranded, let's get this to talk to each other. Here we are. Double-stranded break. You have a 5' to 3' exonuclease activity generating a single strand here, undergoes strand invasion, makes a D-loop. Now you've got a primer and a template. This can be extended. Soon as the D-loop gets long enough, it can now base pair with this other 3' tail. That can then get extended. These pieces get ligated to each other, and magic occurs. We'll talk about that magic. And you end up with a repaired piece of DNA copying from that opposite strand, in this case, the red one. So the really critical thing that happens here is this strand invasion step. So you've got the exonuclease activity. How do you now get strand invasion to take place? Well, there's some very specific proteins that do this. They're called RecA in bacteria. They're called Rad51 in yeast. Why called Rad51? Any ideas? Because they're radiation sensitive. That's what all the geneticists do. They break something and figure out what the phenotype is and then name whatever they found that gene after that. So radiation sensitive. Why radiation sensitive? Ionizing radiation. What does it do? It makes double-stranded breaks. Again, not terribly surprised. So Rad51 or RecA, what that does is it binds to single-stranded DNA, and particularly that 3' overhang that was generated due to the exonuclease. That stretches out this single-stranded DNA, but it also, this RecA, will associate with double-stranded DNA as well. Also stretches out that double-stranded DNA. And then what happens, which is really amazing, is these enzymes, RecA, Rad51, through ATP hydrolysis, switch strands. So they take this gray strand and bind it to one of the strands of the DNA duplex. So this process, the strand invasion, RecA, whatever this protein is, pulls them apart and then puts that strand together with the homologous DNA on the opposite double strand, making a D-loop in the process. And we'll look at this in a little bit more detail as well as we move through here. As I mentioned before, it's not just when you have a double-stranded break that happens through some kind of DNA damage. Turns out that meiosis probably requires this homologous recombination. so lining up the two sister chromatids with each other. You have a endonuclease, SPO11, that gets together with some other endonucleases now, because they're cutting the middle of the DNA. That then gives you a double-stranded break. These ends get chewed in by exonucleases. You have strand invasion, base pairing, and DNA synthesis that happens. This seems to be absolutely required for getting proper meiosis to take place, and that crossing over, and potentially one of the reasons that sex evolved in the first place was by having this kind of recombination that happens between parental chromosomes, so you can get rid of any of the recessive alleles that could potentially cause you lots of problems in the next generation. So this recombination process seems to be one of the real major reasons that you have sexual reproduction. And this is just shown here, where you have a diploid cell, meiosis happens, and then you end up with mostly one parent, in this case the red parent, and a little bit of the blue parent, but also these other little pieces back and forth across these. And we'll look at this process in a little bit more detail a little later on, what happens in terms of these, what happens in some cases, you end' with something called gene conversion. So after you've had this D-loop formation, extension of each of these 3' ends, now you end up with a piece of DNA, a nick, this gets ligated, this piece over here is basically a nick, and we'll look at the structure, it gets ligated, and then you end up with these crossing over pieces of DNA. These are what are called Holliday Junctions, and the way that people always draw them is like this, with one continuous strand here and then these crossing over strands. It's unfortunately not the way that they really look. That's because these strands are wound around each other. And we'll take a look in just a second at what they actually look like. This is actually a completely symmetrical structure that we have here. So it's really 4 strands all coming together in one of these processes. But clearly, if you have base pairing over here and base pairing over here, particularly if you're thinking about meiosis, you try and pull these 2 chromosomes apart, what's going to happen? Actually going to break apart. It's going to be a god-awful mess. And in fact, that's what, again, some of these chemotherapy drugs try and do, is to mess with that kind of process to cause breakage happening. So when you have one of these specific structures, again, a 4-stranded structure all coming together, there are specific enzymes which bind to these structures and cut them in such a way that now the 2 chromosomes can separate from each other. These are called Holliday Junction resolvases, and they can cut either here horizontally or vertically, as they have it shown here. Now, horizontally and vertically here, again, in this 4-stranded structure, look basically identical to each other. And we'll take a look at that in a little bit more detail later on. But the outcome of this process is either what's called a crossover, which takes place. And what I mean by crossover, that means that the DNA at one end of where you had these 2 Holliday junctions is coming from one chromosome, in this case the red one, and the other end is now from a yellow one. And, of course, reciprocally, yellow at one end, red at the other. If, however, you don't have a crossover, and what that means is that both of these Holliday junction resolvases are cut in the same way, now you have what's also called a patch, which is just a short piece where you have exchange of genetic information from one chromosome to the other, but the ends are still the same. So 2 possible outcomes. Because these are symmetrical structures, each of these Holliday junctions, it's, at least in theory, just as likely that you'll have them that cut like this, or they'll cut in opposite ways. So let's look at a little bit more molecular detail what these things look like. As I mentioned before, strand invasion. Strand invasion happens because of the presence of REC-A or RAD51 binding to DNA. This is just a structure of this, I think this one's REC-A protein here in a helix bound to single-stranded DNA. and it's stretching out both that single-stranded DNA and the double-stranded DNA that it's interacting with, and it's through ATP binding and hydrolysis that you end up having this strand exchange reaction, starting with this red single strand and green double strand gives you a red-green strand here, and then a green single strand that comes out the back. Happy with all this stuff? Yes? Do we have our clickers handy? Yes? No. After a double-stranded break in DNA, what is required before strand invasion can take place? 5' to 3' endonuclease activity, 3' to 5' endonuclease activity, 5' to 3' exonuclease activity, 3' to 5' exonuclease activity, or Holliday junction resolution. Ten, 5, 4, 3, 2, 1. What do we got? Okay, endonuclease can't be right. Why? Because that's cutting inside the DNA. So a 5' to 3' endonuclease and a 3' to 5' endonuclease makes no sense whatsoever. So Holliday junction resolution is what? After you've had this crossing over structure that we'll look at on the next slide, and dealing with that. That's way after you've had your strand invasion process. So 5' to 3' exonuclease or the 3' to 5' exonuclease. What end do you need? You need a 3' end. So you want to have 5' to 3' exonuclease activity. So 66% of you got it right. Yay! So C is the correct answer. Okay, so now let's look at those Holliday junctions that I keep talking about in excruciating detail here. So these Holliday junctions, again, this is just a slightly different way of drawing them. Again, because it's a double-stranded piece of DNA. If you just draw the lines here, this would be just like taking those two pieces we had before and pulling them apart. So you crossed over in the middle. But this, because the strands are all wrapped around each other, is actually completely identical to this structure right here. And you can actually see that in electron microscope if you purify these kinds of complexes relative to each other. This is in the electron microscope. And here's a still of an animation that we'll look at in just a second, where you have the double-stranded DNA. So here, blue, dark blue, light blue, and down here, sort of hard to tell, but purple and red, which then exchange from each other. Now we've got red and purple here, and I say red and dark blue, I should say. Purple and your light blue here. So it's really this double-stranded piece, and actually we have a high-resolution crystal structure. It looks more like this, but this I find really hard to actually take a look at. But the idea here that I really wanted to emphasize, if you look at a structure that looks like this, then it's really pretty straightforward to see that here, I'm going to use this arrow here, cutting this way, is basically identical to cutting that way, because again, these are completely symmetrical structures. And what that means is that you can hook up either this piece to that piece, see offset here. So you've got yellow going to yellow, or just as easily you could hook up this piece here, where now you've got yellow going down to red. And again, this is a completely symmetrical structure. So your Holliday junction resolvase can cut and link going one way can cut and link going the other way. And I find the best thing to do with this is just to take a look at it. There's also a number of pretty cool claymations that I found on YouTube. I'll put some links to those in the discussion group. You can actually take a look at how all of these things are associating with each other. My favorite version of this is a fellow by the name of David Rice, who is at University of Sheffield in England, who has actually studied very closely these structures. And so here, it's actually a little bit easier to see. We've got blue and green in one helix, purple and red in the other helix, and this Holliday junction between these. The other thing about Holliday junctions is that they can do something called branch migration. Now, what the heck is branch migration? What branch migration is that basically where these cross structures are relative to the DNA can go back and forth. And how can they do that? Well, they can do that because as you're forming base pairs up here, you're breaking them down here. The same number of base pairs break as get formed, and that ends up making this a completely isoenergetic reaction. You're breaking those hydrogen bonds, you're forming the hydrogen bonds. Just as you break them, you form them as well. So when you have one of these Holliday junctions, it can actually move back and forth on the DNA quite easily. And so here's another example of that. Here we have Holliday junction moving here together with some of these DNA complexes, DNA protein complexes, which allow that movement to take place. Okay, let's switch back over to, yeah.
Will these move in response to a certain enzyme? Are they catalyzed to move? Do they move in response to stress, to strain on the DNA?
So the movement seems to actually be pretty random that's happening here. So they're not really being pushed in one direction or another. You do have, and in fact, the iBiology that I talked about before, there's a nice, get back to this one, don't want that. There's a nice discussion about what happens if these two Holliday junctions run into each other, and actually does occur in some cases, but it's actually pretty rare that they'll run into each other. Usually they're going to be moving in opposite directions relative to each other. But it's this movement, once you form these Holliday junctions, again, right here, this can now move in this direction, can move in that direction, this can move in this direction, move in the other direction. Turns out that this can be thousands of nucleotides that these things can branch migrate in any direction. And so what that means is you're going to end up with quite a lot of sequence where you've got genetic information from one strand paired to the genetic information on the other strand. And if you're thinking about this from the sister chromatid point of view, what that means is, in some cases, these are not going to be identical sequences to each other. So you have one strand that has one nucleotide on it, the other strand that actually has a different nucleotide on it in that position. And so that's what leads to gene conversion. Now, that's because, and these are actually relatively short, where they've shown these sequences with branch migration. Some of these are actually going to be really quite long. Here, this is just looking at a single chromosome after you've had meiosis and after you've had these double-stranded break repairs, which has given you either a patch, like you have here, or a crossover, as you have here. So if you happen to have sequences in here which are not completely identical between your maternal and paternal chromosomes, what happens? Well, what happens is you have mismatches. What happens when you have a mismatch? It causes a change in the structure of your DNA. What happens if you have a change in the structure of your DNA? That gets repaired, gets chopped out and repaired. Now, the difficulty here is, unlike replication, we had Nicks on one of the strands, that one's the new strand, you've got methylation on one strand versus another one. It does happen a little bit in E. coli, this kind of homologous recombination, less frequently, but can happen. But much more so in the diploid replication. So you've got these two strands, and one of those mismatches has to be repaired. But the mismatch repair machinery doesn't know which one's the right one and which one's the wrong one. It's not that dad's right and mom's wrong. Usually mom's right, at least in my experience. So, but the DNA repair machinery doesn't know that. And so what it does is it will cut out one of these strands, pardon me, and have a replacement that happens in there. Oh, come on. So here, cuts out and does a replacement. What does this mean? Well, if it turns out that there's an allele here. Allele mean things to people? Yes? Genetics? No? Allele? What the heck? So hopefully allele means something to you. So genetics is going to be a difference between maternal, paternal. So if it turns out that you have a difference right here in the alleles from the maternal and the paternal, if one of those has been repaired, you can end up with gene conversion in such a way that you don't have nice Mendelian genetics that takes place. You know, AA, little a, big A. You could actually end up with more copies of one of these genotypes than you would expect due to normal segregation. And this happens really quite frequently. It happens even more frequently if you have more of these double-stranded breaks and more double-stranded breaks that get repaired. So this is something originally discovered in yeast, but it turns out happens with not really high frequency. And again, getting back to your point about where all these double-stranded breaks happen, coding sequences, less coding sequences. So it's not going to happen that frequently in vertebrates and in us, but it still does happen with not unknown frequencies. And it really has to do with this branch migration process, where you end up with these two different strands from the different parents. So any questions about the double-stranded break repair? No, the next slide is not a clicker question. You've seen it already. Okay. Last thing I wanted to talk about in terms of DNA repair has to do with what happens if stuff goes really, really wrong and you still have to replicate through something. Yeah. Sure. Yeah. Mm-hmm. Okay. So here, the question is how does it get fixed? Basically, it gets fixed just like normal mismatch repair would be. And the template here is now the red. So the red genetic information is what gets passed along. If, however, the red strand had been cut out, it would be the blue information that gets copied. I was just thinking about genetic information in this case. But yeah, it's just copying from that other strand. Because the bases here are totally normal. There's no difference between the two. They're just mismatched because mom had an A there and dad had a T there. Okay, yes, guys. So any connection between dominant and recessive alleles as far as selectivity is concerned? The answer is no, because that's all the selection is going to be happening at the phenotype level. We're talking here at genotype. The DNA repair machine doesn't have a clue what this DNA does. So yeah, it makes no difference of dominant, recessive, etc. Yeah, we're not talking genetics here. This is the DNA repair machinery has no clue. Okay, so if you end up in a really nasty situation, and this does happen, particularly under stressful conditions, like say last Wednesday, cellular stress can lead to lots of DNA damage, and part of that actually has to do with the upregulation of reactive oxygen species. Reactive oxygen species change a lot of chemistry. Change a lot of chemistry can be a big problem in your DNA. So what happens under those kinds of conditions is been best studied in E. coli, but it turns out also happens in mammalian cells, eukaryotic cells, is there's activation of recombination machinery. So helps with Rec A. Rec A is your binding to those filaments, helps you repair some of these changes that take place. A number of other genes are expressed, particularly blocking cell division, because if there's a lot of DNA damage that's happened, you don't want to be replicating your DNA and trying to separate your chromosomes before you've had a chance to repair those kinds of things. And these things called error-prone or translesion polymerases are being expressed. You make these new DNA polymerases. So what do these new DNA polymerases do? Well, what they do is they will bind to your sliding clamp, again, beta and E. coli, pCNA and eukaryotes, and will bind to these specifically where you have what's called a stalled polymerase. So this is your normal replicative polymerase, the green one here. Gets to some kind of DNA damage that hasn't been repaired, hasn't been dealt with yet. So generally, this will get repaired and you can continue on to replicate it. But if you've got a whole bunch of DNA damage, probably due to some cellular stress which has happened, you still need to replicate this piece of DNA, and the idea is basically you just want to repair it later. We'll come back, we'll deal with this later. But to do that, you somehow got to get your 3' end past this piece, up to the piece of template that you continue to replicate on. So here you get one of these so-called translesion polymerases or error-prone polymerases that comes on, binds to pCNA, puts in a couple of nucleotides, and then falls off. But the key here is these are error-prone polymerases. They'll put in whatever nucleotides in that position. And so actually can often introduce mutations at that particular process because they're relatively random nucleotides. Fortunately, they only got 4, and so most of the time you'll be right if there's a couple of nucleotides that are bad, but sometimes it won't be. And so you can actually end up with a number of mutations that happen. It's quite well known, again, in E. coli. Here's your beta clamp. Here's DNA polymerase 3. Gets to a thymidine dimer, which is normally repaired through what process? Nucleotide excision repair. Then the sliding clamp actually helps bring in one of these translesion or error-prone polymerases. This puts in a couple of nucleotides. It falls off. You have reassociation of the replicative DNA polymerase and continue. This is actually one of those translesion polymerases here. All of you can see the right-hand structure. I can't. Bound to DNA, and this particular modified DNA that it's replicating past. So it's getting really bad. You still have to finish your replication. Get some of these error-prone polymerases, which are not processive. If they were processive, it'd be a real problem. You'd be putting in wrong nucleotides all the time. Just to get you past that particular lesion so that you can continue to replicate your gene. So, last time we talked about some of the human diseases. I talked about them to my computer. Many inherited human diseases, and particularly true in lots of cancers, are due to changes in a lot of DNA repair genes. So if you're deficient in some DNA repair genes, the classic example is xeroderma pigmentosum, hypersensitive to sunlight. The reason they're hypersensitive to sunlight is they're missing some of the proteins in the nucleotide excision repair pathway and end up developing lots of cancers because these things don't get repaired. There's a little bit about the mutations, our specific repair. Proofreading you can also think of a little bit as being repaired because the wrong nucleotide gets put in. Mismatch repair. Didn't talk too much about photoreactivation, which is a smart way to deal with pyrimidine dimers. Just reverse the reaction. Unfortunately, we don't have this enzyme. It'd be really cool if we could actually express this particular protein, particularly in people who have xeroderma pigmentosum. Great science fair project? No. Thinking about these kinds of modifications. And then today we talked about the base excision repair, nucleotide excision repair, but mostly about double-stranded break repair. One thing that I forgot to mention, there was also another thing that I found online. There's a really nice review of non-homologous end joining that I found that I'll also post on D2L. So let's finish up, just like everyone wants to, with a clear question. Which of the following DNA repair mechanisms are most likely to cause gene conversion? Mismatch repair, base excision repair, nucleotide excision repair, non-homologous end joining, or error-prone polymerases? Thank you. So, half and half between mismatch repair and non-homologous end joining. Non-homologous end joining can cause mutations, but it's not causing gene conversion. The gene conversion is going from one allele to a different allele, as opposed to just causing that particular kind of mutation. So the correct answer here is A. And we could talk about transposons, but yes, no, no thanks.